what used to be a big puzzle. And the way that's done is you have DNA, which is a double helix, and it gets copied to a single strand of RNA, which also consists of 4 different bases. And now you have to go from here to the language of protein, which is a chain of amino acids. And the way that's done is groups of 3 bases are read to code for an amino acid. And Crick correctly predicted that there would be an adapter molecule, and that's called tRNA. And what tRNA has, this is simply a cartoon of tRNA, it has 3 bases that base pair with the triplet, with a group of 3 bases on mRNA, just like the bases base pair within the double helix. So it's the same kind of chemistry of base pairs that occurs in the double helix, but this time it's between tRNA and the messenger RNA moiety that carries the information. So you can see that a tRNA comes in, it recognizes its triplet in pairs and brings along its correct amino acid. And then the next tRNA comes in and it brings in its amino acid, which is different, or it could be the same, but potentially different. And then these 2 amino acids have to be joined together. And then the process has to continue along this genetic message. So essentially we're taking genetic information present in our genes, and using that information to make chains of amino acids, which are proteins, which fold up. Now, clearly, this doesn't happen by itself. And that was realized very early on when people who were looking at where newly made proteins were in the cell by labeling using radioactive amino acids and doing electron microscopy, they showed that newly made proteins localized on these particles in the endoplasmic reticulum. And these particles were isolated, and they contained, they all were about 250 angstroms in diameter and contained 2 subunits, a large subunit and a small subunit. And it turned out that all species of these particles from all species consist of a large and a small subunit. And these particles were called ribosomes. Now, most of you know Jim Watson because of his discovery with Francis Crick of the double helical structure of DNA. But it turns out that his lab actually pioneered some of the early work on ribosomes. But because he didn't actually do the work himself and didn't actually write the papers, but he allowed his students and postdocs to work very independently, he didn't put his name on any of the papers. Now there's a novel concept that perhaps, big, Indian scientists might take note of. So here he is talking about the early days of ribosome work. Oh, 1 minute.
A little round object doesn't contain DNA, but contains carbon. And I started studying it with Alfred Tischer, here at Harvard, and we knew that it was involved in on the ribosome. A little round object doesn't contain DNA, but contains carbon. And I started studying it with Alfred Tischer here at Harvard in the spring of 1967. And we knew that it was involved in how proteins are made because the newly made proteins were tested. So we thought if we solved the structure of proteins of the ribosome, we would know how proteins. And that was a big puzzle because you had to somehow go from DNA to information, DNA to information perfect. And there was this particle which we thought was round and circular. Hopefully only one sort of protein. If it did, then maybe it would not be easy to solve, but it would be solvable. So over the next couple of years, after I assembled some people, first the protein chemist who looked at it said, I'm leaving. And because it wasn't simple. And then a protein chemist actually said, gee, there's these dirty proteins in this world out there. And so automatically I knew we'd never know its structure.
So it is true that in Watson's time, in the late 50s, it would have been impossible to determine the structure of the ribosome. But it didn't stop a large community of biochemists worldwide from characterizing the ribosome because it was so important. And they first figured out what the ribosome is made of. There are about 50 proteins and 3 pieces of RNA in bacteria. And then they showed how the subunits would assemble. And this is what an electron microscopy image of the ribosome looks like. And then about 40 years of biochemistry showed that the ribosome, the small subunit binds to the genetic message, and the ribosome has slots in it for these tRNA adapter molecules. And the way that happens is the first amino acid binds, and then the second amino acid binds in the second slot, and that enables a bond to be formed, which perhaps Tom will talk about in his talk. And so you get a bond between the first and second amino acid. And then the whole assembly has to move, and so that results in the first tRNA being ejected, and then the ribosome assembly is moved to the next triplet, and then the third tRNA comes in, and then a bond is formed, and so on. So you can see from this picture that the message, the information is being read here, while the polypeptide chain of amino acids is being synthesized over here, and that's done by this large, complicated machine called the ribosome. Now remember that my cartoon picture, this is a highly simplified picture of the ribosome, and the first breakthrough was in understanding what this tRNA molecule looks like. And actually it doesn't look like this, it's simply a conceptual cartoon. It looks like this, it's L-shaped, and here's the anticodon triplet that base pairs with the triplet. So this base pairing actually occurs here, and the amino acid is brought here. Okay? And this is the view of the ribosome we had in the kind of early 90s, where here's the small subunit, and it binds the message, presents the triplet, the L-shaped tRNA binds the codon here, and the amino acid is brought here to an active site where peptide bond is catalyzed, and the nascent peptide emerges through a tunnel in the ribosome. Now, this was, of course, tremendous progress from knowing nothing about the ribosome to knowing a lot about it. But if you want to understand the nature of the ribosome in chemical terms, and if you want to understand how antibiotics bind it, you need a high-resolution structure of the ribosome. And to do that, you have to understand how we normally visualize small objects and see detail in small objects. The way you do that is, if you have a small object and you want to look at it in detail, typically you would use a lens. And what a lens does is it collects scattered rays from the object and recombines them. And if you use your lens correctly, the image will be magnified, and then you can see that with your naked eye. Now, in fact, all of you are using lenses to make an image right now when you're seeing the screen, because the light scattered from the screen is going all over the auditorium, and you're taking a portion of that light, and you have a lens in your eye that recombines the rays and forms an image on your retina. That's why you're able to see this image. However, there's a law of physics that says that you cannot distinguish detail that's smaller than the wavelength of the light that you're using. And the problem is that light has a wavelength of about 5,000 angstroms. And the distance between atoms in a molecule is typically about 2 angstroms. And so you cannot using light, ordinary light, use a magnifying glass or a microscope in order to discern details of the atomic level. You can use a technique called, you can use light of a much shorter wavelength, and if you use light of the appropriate wavelength, which is 1 to 2 angstroms, those are X-rays, okay? So X-rays are simply photons, just like light, but they're much shorter wavelength. The problem is that there's no lens that will actually recombine the rays like an optical lens. There are lenses developing now, but they don't have sufficient, they're not there where you can look at individual molecules. The second problem is that X-rays are damaging. So if you look at a single molecule, just like you would look at an object using a magnifying glass, the amount of X-rays you would need to get enough signal from a single molecule would actually destroy the molecule. So even if you had a very powerful lens, you wouldn't be able to get, using continuous X-rays anyway, an image of the object. But there is a technique that you can use to circumvent that, and that's called X-ray crystallography. And in X-ray crystallography, what you do is you crystallize your object, which means you form very regular three-dimensional arrays of the same object, like the ribosome. And you take that object and you put it in an X-ray beam and you hit it with X-rays, and the X-rays are scattered, just like any light is scattered from an object that it hits. Okay, so X-rays are scattered, but because it's a crystal, it has two consequences. Unlike light rays, which are scattered in all different directions when they hit an object, when you have a crystal, the rays are only scattered along certain directions because of only those directions have what's called constructive interference. The other thing that happens is that the signal from all the millions of molecules in the crystal is added so that you get enough signal even from these macromolecules, which are small by our standards of everyday standards. And so you get enough signal to measure the scattered rays. And what these scattered rays look like is like this. This is an X-ray image, a detector image, and you can see the scattered rays are in these spots along the detector. And what the process does is it takes these scattered rays and recombines them into an image, and that produces a three-dimensional image of the object. Remember, whether there's a lens there or not, the scattered rays are present from an object. So even if you are not sitting in your chair, the scattered rays from the screen or me would actually hit you, hit your chair. And the fact is that you have a lens that collects the scattered rays and makes an image. So you can do in a computer what a lens does in real space, which is to collect these scattered rays and recombine them to make a three-dimensional image. Now I'm simplifying the problem because there is a problem called the phase training, which I won't go into. But that's fundamentally what we do. We recombine scattered rays in a computer to produce an image. Now, in order to do that for the ribosome, you need crystals of ribosome, and the first breakthrough was made by Ara Yonat, who was here yesterday and produced crystals of the 50S, and then a group in Russia produced crystals of the 30S in the entire ribosome, and then a number of us had to contribute in order to eventually get a structure. Now, one of the ways that, one of the things we needed contribute in order to eventually get a structure. Now, one of the ways that, one of the things we needed were these very, very powerful sources of X-rays, because ribosomes are very large molecules in molecular terms. And so the crystals of ribosomes don't diffract very well. That is, the spots are not very intense. And so you need an extremely intense source of X-rays. And these are some of the synchrotrons we use. And this one is in France, these two are in the US, and this one is in Switzerland. So I like to joke that you can join a ribosome lab and see the world. This is a paraphrase of the US Navy's recruiting campaign, which used to be join the US Navy and see the world. But of course, these people are not actually going sightseeing. They're stuck inside a ship most of the time, And of course, my people aren't going sightseeing either. They're actually inside the synchrotrons all the time. So they're only seeing the world's synchrotrons, not actually seeing much of the world. And what they're seeing is something like this. This is the inside of a synchrotron instrumental touch. And this tube brings the X-rays from the synchrotron ring, and this is where the crystal is, and it's cooled. It's kept cooled by a flow of nitrogen at very low temperatures. And this is a CCD detector, which measures the scattered X-rays. So when you finish this experiment, and you've done it correctly, what you end up with is a three-dimensional image of the object you're looking at. And in our particular case, originally it was the 30th subunit of the ribosome. And this is what it looks like. Of course, the problem is this doesn't tell you what the atomic structure is. This is simply a three-dimensional image of the object, although in much greater detail than you would see otherwise. So how do you go about from this three-dimensional image to an atomic structure? That's a bit like solving a complicated jigsaw puzzle. But there are fundamental differences between an ordinary jigsaw puzzle and this kind of jigsaw puzzle. First of all, this is in 3 dimensions, not in 2 dimensions. The other problem is, when you do an experiment, there are always errors, and you don't know what those errors are. So you may see images where there shouldn't be an image. You may see part of an image where there actually should be empty space. Or you may see vice versa, there should be something there, but you don't see it, either because it's moving around, so it's blurred, or because of errors in your measurement. So it's like solving a jigsaw puzzle where the shapes don't always fit correctly. And you have to use knowledge to realize, okay, this is a region that isn't fitting exactly, and this is a region that fits exactly. And that takes a lot of prior knowledge. Of course, the most important difference is that when you buy a jigsaw puzzle, there's a picture of the puzzle on the cover of the box. So you already know the answer before you start solving the puzzle. Whereas here, we don't know what the molecule looks like, okay? And that's perhaps the most difficult thing of all. So what you do is you start looking for regions you can recognize. Just as in a puzzle, you would look at the corners and the edges first and build those, and then work your way into the more and more difficult parts. Here, what you'll do is you'll zoom around your object and see what you recognize. And if you zoom in, what you can see is here's a region where there's a regular repetition of these bumps. And on the other side, you can see an equally regular repetition of the bumps. And if you know your nucleic acid structure, you'd recognize these as phosphate groups connected by sugars. And in between these two strands would be these bases that are interacting with each other. And so what you would then do is build into the molecule, and you would thereby explain a piece of that image in atomic terms. You would interpret it in atomic terms. So what you've done now is taken that image and interpreted it in terms of an atomic structure, and that's what that structure would look like. But remember, this is only a small part of the entire image. You've built a small part of the whole image, and so what you then do is keep on building all of the parts you can recognize until you've accounted for all of the image. And so that's what you do. First you ask, where is the part that I've built in the context of the whole image? And then you would gradually start building the entire image until you've accounted for all of the image. And that's when you realize you've actually solved the atomic structure of the molecule, okay? And that's a very special moment in a structural biologist's life. So using that, Tom Stites' group at Yale had solved the structure of the large, or the 50th subunit, while we had solved the structure of the small, or the 30th subunit. And this actually shows that because the ribosome consists of both protein and RNA, it's about 2/3 RNA, And one of the most important things was it gave us a glimpse into what an early ribosome might have looked like, because today's ribosomes pose a dilemma. If the ribosome is used to make proteins, and the ribosome itself consists of proteins, where did the ribosome come from? Because if it's the thing that makes proteins, how could it be that it also contains proteins? Well, Crick long ago suggested that maybe early ribosomes consist only of RNA. And even today's ribosomes, the active site of the ribosomes, where the peptide bond is formed, or where the tRNA codon is recognized, anticodon recognizes the mRNA codon, consists entirely of RNA. The proteins are not involved in these very critical regions. And that strengthens the idea that an early ribosome may have consisted only of RNA and proteins got added later. Now here is a picture of the entire ribosome that we saw in 2006 with the tRNA and the mRNA ligands bound to atomic resolution. Now this brings me to the idea of how you could recognize antibiotics. I started off with antibiotics and how they bind to the ribosome. The point is that once you had a structure of the ribosome or its subunits at high resolution, you could actually then very straightforwardly determine the structure of an antibiotic bound to the ribosome. Because if you took the data from the antibiotic bound ribosome and subtracted the image from the free ribosome, what would be left is the image from the antibiotic alone. And I show you that over here. This is the image from the antibiotic alone, and that's obtained by using a difference technique. And you can see that this is interpreted now in terms of the antibiotic spectinomycin. And spectinomycin is very small compared to the entire 30S subunit, but the reason it can work is it blocks a very critical hinge point between the so-called head of the ribosome, 30S subunit, and the body. And this head is known to move with respect to the body. And so by binding at a very crucial point, even a very small molecule can block the function of a large thing like the ribosome. Now, there's a very clinically important antibiotic called tetracycline, which binds over here. But it turns out this is exactly where those tRNA molecules that bring in the amino acids bind. So if you have tetracycline here, it won't be able to bind to the ribosome, and that's why tetracycline stops protein synthesis. Now I want to close with a couple of things. One is, why do bacteria develop resistance? Well, there are several ways that bacteria can develop resistance. You can see that antibiotics work by having a precise pocket in which they bind to in a target molecule. So these ribosomal antibiotics will bind to the ribosome in a very precise pocket where they fit exactly. Now that kind of fit is necessary for the antibiotic to work. But you can break down that fit in several ways. One is this bacteria can make proteins which simply break down the antibiotics. If they break down the antibiotics, then they no longer are intact and can bind in a pocket in a specific way. The other thing they can do is have proteins that modify the antibiotic by adding groups. A typical group that they add is an acetyl group. So you have acetyl transferase enzymes which add chemicals to the antibiotic. And because they've now changed the shape of the antibiotic, it doesn't fit in the pocket properly. A third very common mechanism is that you have pumps in the bacteria that recognize foreign substances like antibiotics and pump them out of the cell so the antibiotics don't do any harm to the bacteria. So these are three completely different ways of acquiring resistance. The one thing that's guaranteed is that if you use an antibiotic, eventually bacteria will become resistant to it because of natural selection and selection pressure, which is very powerful. And so it's very important to use antibiotics wisely. If you want to develop a new antibiotic, and Tom Stites can tell you this much better than I can, because he founded a company called Rydex to develop new antibiotics, and perhaps you'll talk about that. To develop a new antibiotic costs over $1 billion because you have to do a tremendous amount of screening to identify new compounds. You have to be able to synthesize them, test for toxicity, for side effects. You have to see that they're actually working inside a human body. It's all a huge amount of work. It takes up hundreds of people's time for many, many years before something actually can be used in patients. So it's important to treat infections in other ways. One is you have to promote rational use of antibiotics. I notice in India, whatever the laws are, you can go down to a chemist or pharmacist or medical supply store and say, I want this antibiotic, azithromycin. And without questions, the guy will give you azithromycin, regardless of whether it's a proper antibiotic or not. Okay? Your friend might say to you, oh, I was sick and I took azithromycin and it was perfectly fine. So, next time you're sick, you should take that. That's not a good way to use antibiotics. Antibiotics should be highly regulated. And they should not be given for diseases that they are not effective for. Some antibiotics are better against certain diseases. They're useless against viral diseases. So you have to be careful about the use of antibiotics. The other thing you need to do is surveillance. One of the important things is to make sure that you identify TB victims, TB patients, people who have TB, and make sure that you've identified them. That takes public health and surveillance. If they have other diseases, for instance, elephantiasis is a bacteria. Elephantiasis is caused by a parasite, but the parasite has a symbiotic bacterium which is sensitive to doxycycline, which was recently discovered to be effective against elephantiasis. By killing off the bacteria, you indirectly kill the parasite. And doxycycline is also used as a malarial prophylactic, and it turns out it binds to the 30th subunit because it's a close analog of tetracycline. So again, if you identify, but it doesn't work against the later stages of elephantiasis where you've already done damage to the lymphatic system. So you have to identify when people have elephantiasis at an early stage, and that then takes surveillance so that they can be treated. You have to use infection control. This involves sanitation, prevention of, if you have an epidemic, you try and stop the infection. So those are all public health measures that are far more effective than developing new antibiotics because they're much cheaper than having to develop new antibiotics. But of course, we have resistant bacteria, so we always have to stay ahead of the game, and we need to develop new antibiotics. And that again has many aspects to it. We need, if you have an epidemic, you try and stop the infection. So those are all public health measures that are far more effective than developing new antibiotics because they're much cheaper than having to develop new antibiotics. But of course, we have resistant bacteria, so we always have to stay ahead of the game, and we need to develop new antibiotics. And that again has many aspects to it. We need to understand microbial pathogenesis. We need to understand how bacteria are actually, why certain bacteria are virulent, what is the cause of virulence, and how they interact with the host. And that will give us more targets to kill off bacteria. And then we need to have better diagnostics that will also help with surveillance. We can try and develop better vaccines as a preventive measure. And finally, with structures like the ribosome, you can develop new drugs and novel therapies for bacteria. So I've told you a lot about the ribosome, and I want to close with a movie of the ribosome to show you how complicated the process is, to give you a realistic understanding of what the process is. And of course, there's a lot about it that we don't understand in atomic terms. So I'm going to close with this movie, and that's shown here. So here is the genetic message in the form of mRNA. and this is the small subunit. And there are 3 protein factors that help guide it to the start of the message, along with some base pairing between the RNA and the subunit itself. Now these protein factors eventually help with the joining of the large subunit and leaves, and this factor called IF2 eventually hydrolyzes a molecule called GTP and leaves, and that leaves the ribosome with the first tRNA and the first amino acid. And then what has to happen is a protein brings a new tRNA to it, and it eventually leaves, and now this second tRNA brings an amino acid that's joined with the first amino acid in the large subunit of the ribosome. Now these tRNAs move first with respect to the large subunit, and then with the help of factor, they move with respect to the small subunit and drag the genetic message with them. So now there's a new triplet on the mRNA that's waiting for the new tRNA that comes in and binds and brings the third tRNA to the ribosome. So you can see how much more complicated it is than that early cartoon version I showed you. This happens at the rate of 20 residues per second, and the polypeptide chain of amino acids emerges from the ribosome through a tunnel and folds into the protein. The shape of the protein, the structure of the protein depends on what the protein is, which depends on what the sequence of amino acids is. And eventually it reaches the end of the message, where it reaches a codon called the stop codon, and that's recognized by a factor which cleaves off the protein, so the protein floats away from the ribosome after some processing. And then another factor, the protein factor, splits apart the ribosome so it can start all over again on either the same or a new message. So I hope to have had some idea of how proteins are made, how antibiotics work, what went into solving the structures, and so on. So you have a rough flavor for what this field was about and why it got the Nobel Prize last year. Thank you very much.